So here is the second of two videos on passive transport. So we left off discussing simple diffusion and then facilitated diffusion. And so the last of the three types of passive transport is osmosis. Okay, so we hear a lot about osmosis, sort of just in everyday culture, I think. But what is osmosis in terms of science? Well, osmosis is basically just describing specifically how water moves across a membrane that is selectively permeable. Okay, so there is some terminology around that hopefully we'll try to sort out. But, as with all diffusion, we're looking at the molecules in question moving from an area of high concentration to low concentration. But that's a little bit awkward with water, moving from high concentration to low concentration. So we're going to break that down for you. So again, when we talk about high concentration to low, we're talking about it moving with the concentration gradient or along a concentration gradient. Okay, so just to take a look at this visual here, right? So just imagine that we had a sugar solution over here on the left, right? And so you can tell that it's more concentrated on the left side of this beaker than on the right, and it's separated by a selectively permeable membrane, right? And so the reality is that the sugar solution, the sugar molecules, are probably too big to pass across this membrane, right? And on the right-hand side over here, we have a relatively more dilute solution of sugar. And we can tell that because I can see fewer dots here, which are each representing the sugar molecules, right? So again, this membrane here is separating the concentrated solution from the the highly concentrated from the low concentration solution of sugar. Could be any solution, but we're just using sugar here. And so what's going to happen? Well, the molecules of sugar can't move. They're too big. But what can move are the water molecules. And that's exactly what happens. And so the water is going to move in the direction that's going to try to balance out the concentration so that the concentration on the left side of the beaker is the same as the right. So water has to move out of the dilute solution and into the concentrated solution. And so what you see as a result of that is the volume of water on the right becoming less and less, and the volume of water on the left becoming greater and greater. And that's just, again, because it's only the water molecules themselves that are moving and not the sugar. So when the concentration of solutes dissolve, those are the dissolved substances, the substances dissolved in water, is lower in the solution than inside the cell. So we're talking about out here, right? So low concentration out here, and high concentration inside the cell. So when the solution concentration outside the cell is really, really low compared to inside the cell, we say that that solution is hypotonic. Okay? So, well, what's going to happen? Well, imagine that the particles inside the cell are too big to move. Well, what happens is that the water wants to move into the cell to balance out the concentration. So, well, what happens? Well, the cell itself usually gets very, very large. It gets larger in comparison. And I'm going to try to make this look even, but then we end up with equal concentrations after diffusion happens inside and outside of that cell. And so what happens as a result, sometimes if the cell gets so much water, sometimes we see the cell actually burst and die. So in that situation, that's not really good. So it's actually the reason why when you're receiving IV fluids, the fluids aren't water themselves. There's some sort of solution that has a balance of dissolved materials compared to the water. So like a salt solution, a saline solution. Sometimes it's like a sugar solution. But either way, you want to make sure that the solution is not pure water, because an infusion of pure water into your cells might not be healthy for your cells. That would cause the cells to possibly burst. So again, that's a hypotonic solution where the concentration of solutes is higher inside the cell than outside, and so water wants to move into that cell. So let's look at the opposite situation here. So an alternative to that is that you have hardly any solutes inside the cell and tons outside. I'm going to try to make this look pretty decent. So just imagine lots and lots of solutes outside the cell. Okay, so you're like soaking your cell in like a salt solution. Well, what is going to want to happen? Well, the water inside the cell is going to want to move out of the cell in order to balance the concentration, in an effort to balance the concentration in and out of the cell. So what's the result of that? Well, you're going to see your cell kind of shrivel up. Okay, so instead, and inevitably, you know, if the cell doesn't die, you're going to end up with sort of an even concentration. At least that's the goal of solutes inside and outside the cell. Again, this is not, you know, meant to be accurate. It's just to give you an idea. And so what do we call that situation? We call that a hypertonic solution. Okay, so the solution is hypertonic, and the water is going to move out of the cell when placed in that solution. So the last situation is a situation where we have an isotonic solution. And so in that case, we have an equal number of solute particles inside and outside the cell per unit volume. Okay, so that means equal concentrations inside and out. And so it's not as though there's no movement of water in this case, and this is really important to understand. It's simply that we've reached an equilibrium of sorts, so that for every water molecule that we see moving into the cell, we will also see another water molecule trying to balance that situation by moving outside of the cell. That's the ideal situation for animal cells. But we're going to talk a little bit more about what that means for plant cells. So here's a nice chart that compares animal cells to plant cells and how they manage different types of solutions vis-a-vis osmosis, so hypotonic, hypertonic, and isotonic. So you'll notice on the top here I have the situation for animal cells. So an animal cell in a hypotonic solution, that's the case where water is moving into the cell because the concentration of solutes is too high, then we're possibly going to see that cell lysing, okay, or bursting. That's essentially what that means. So like I was just saying in the previous slide, an isotonic solution is the ideal situation for our cells because, you know, for every one water molecule in, we have one moving out, and so it maintains a consistent sort of size and shape. A cell will shrivel, though. An animal cell will shrivel in a case of a hypertonic solution. Why? Well, remember that water is trying to move out of the cell to balance a high concentration of solutes outside. And so the cell becomes shriveled and can die as a result. Right? So either of these extremes are not good. We want an animal cell to be in an isotonic solution. Plant cells are a little bit different. Remember that plant cells have a cell wall that can prevent the cell from bursting, like in the case of an animal cell. And so what's normal for a plant is for the plant to be in a hypotonic solution. Right? We want water to be able to move into that large vacuole and keep that plant nice and turgid, okay, so or firm, right? Think about if you were, you know, I don't know if you do this at your house, but at our house, if the celery is getting kind of old, then we cut the end off and we just place it in some water for a bit and it'll firm right up. It's a nice way to make crispy French fries too. If you take potatoes that are kind of a little bit soft and you put them in water, they'll firm up and make your French fries a little crispier. On the opposite side of that, You have the hypertonic solution. The hypertonic solution is very, very bad for plants. If you put them in salt solution, they will shrivel up and completely die. It's why that the salt on our roads in the wintertime is a big concern environmentally, because it can kill plant life that's sort of on the side of our major highways. Okay, so you'll still get in an isotonic solution with plants, you'll get the plant looking flaccid. It won't be dead, but it'll be floppy, and that's not really where the plant wants to be. We want the plant to be turgid. It has that cell wall that will help to maintain its structure and prevent it from lysing. Okay, so a quick comparison of plant versus animal cells and how they manage osmosis. So again, ideal for an animal cell is an isotonic solution. Ideal for a plant cell is a hypotonic solution.